today's class deals with sex determination, sex linkage and genetic disorders. We will be answering several of the MCQs which have been coming in competitive examinations. So we go with relation to question number 1. It states the terminology, the term chromosome was coined by, is it A bit is Hofmeister, B is Sutton, C is Boveri and D is Waldeyer. The correct answer for 1 is D, there is Waldeyer. Now Waldeyer is always associated with the discovery of the term of chromosome. He coined the term chromosome. Chroma means colored. While if you take Hofmeister, the choice number A, Hofmeister is associated with blood groups. While Sutton and Boveri are associated with, Sutton and Boveri, choices number B and C, are associated with the chromosome basis of inheritance. That is, they are associated with the concept of the chromosome as the vehicles of inheritance. Chromosomes are the vehicles of inheritance, or they are the carriers of the DNA. They are the carriers of the DNA. Thereby, for question number 1, the correct answer is Waldeyer. D bit is the right answer, which says Waldeyer is always associated with chromosomes. We are going to question number 2. The question number 2 says, where are chromosomes and what are the chromosomes which are present in the salivary glands of Drosophila called? If you take Drosophila melanogaster, is commonly called the fruit fly, or it's also termed as the Cinderella of modern genetics. The person who has studied Drosophila melanogaster is T.H. Morgan. So when you take Drosophila melanogaster, in Drosophila melanogaster, we have the presence of polytene chromosomes. Polytene chromosomes are always associated with Drosophila melanogaster. Hence, the correct answer is B. Parallel to this, we will also be looking at what are the other options. When you say polytene, polytene means many gene transcripts. So, when you take polytene chromosomes, there are many gene transcripts, and if you take each of the transcripts could be made up of an intron as well as an exon, coding and non-coding sequences. While we have even the other choices are, we also have lamp brush chromosomes. Lamp brush chromosomes are common in the case of amphibian oocytes, and similarly we have even supernumerary, that is extra chromosomes which are present in maybe some of the features of the insecta. And B chromosomes, we have primarily B chromosomes. B chromosomes are also called accessory chromosomes. Accessory chromosomes are present in humans. In humans, an extra bit of a chromosome is called a B chromosome. So for question number 2, The right answer is polytene, and the answer is B. Answer is B. Primarily for chromosomes present in the salivary glands of, primarily in the case of the salivary glands. When you take the salivary glands, now we go to question number 3. Question number 3 says, what is a chromatid? A chromatid, part A says, is it 1/2 of the chromosome, or is it the haploid chromosome, or is it the complete chromosome? Otherwise, is it a duplicate chromosome? As you understand, when you take chromosomes, chromosomes are rod-like structures which are clearly seen during the cell division of a living organism. And when you take a chromosome can be segregated into 2 halves. The 2 halves are predominantly termed as chromatids. And when you take the chromatid is 1/2 of the chromosome is termed as a chromatid. Hence, the right answer is A. So, chromatid is the coloring or the staining half component of a chromosome. It is a coloring, staining of the half component of a chromosome, and thereby it is made up of finer threads which are termed as chromonymata. The chromonymata are condensed and they form, if they are expressed in the form of rod-like particles, then they are predominantly termed as chromatid. The right answer for question number 3 is A, that is chromatid. Now we are going to the question number 4. Question number 4 states a chromosome with subterminal centromere. Subterminal centromere, that is, we are talking about the centromere. The centromere is a basic unit which divides the chromosome into equal halves. So we are looking at a terminal centromere. Terminal means which is present at the end, but it is below the end. So thereby we have a chromosome which is subterminal. So this status is termed as subterminal, just like a submarine. If this is the terminal end of a chromosome, we have this position of the centromere is just little behind the terminal end. So this sort of a centromere is predominantly termed as acrocentric. It is termed as acrocentric. Acentric, the other choices are acentric. Acentric means without a centromere. Acentric means no centromere, acentric. Whereas metacentric means at equal planes. Metacentric. Then later on we also have a heterocentric. Telo refers to terminal end. So it is not a heterocentric chromosome, it is not a metacentric chromosome, it is neither acentric. So hence the correct answer is B, which refers to acrocentric chromosome. In question number 5, it's asked, what are puffs or Balbiani rings in the salivary gland chromosomes? What are they? Are they sites of DNA replication? Are they sites for DNA duplication? Are they sites of RNA synthesis, or are they sites of protein synthesis? Now, when you take chromosome puffing, the meaning of the term chromosome puffing is chromosomes are metabolically active. So when they are metabolically active, the DNA is activated. So while the DNA is activated, puffing has been discovered by a person whose name is Balbiani. Hence, it also goes to form Balbiani rings. Balbiani rings or chromosome puffing, and this sort of an extension, this sort of a character of puffing is normally seen in the salivary glands. Is seen in the salivary glands of the chromosomes of insects. So these, when you see they are actively, DNA is activated, the choices are they the sites of DNA replication? Now they are not the sites of DNA replication because a DNA does not make a DNA. So this is DNA replication. DNA duplication, DNA doubles itself. So even the second choice is wrong. And it's not predominantly during chromosome puffing, there is no formation of RNA. There is no formation of RNA along with protein synthesis. Along with protein synthesis. So the information flow from DNA to RNA to protein synthesis is not there. Hence the choice D is wrong. Whereas in chromosome puffing, predominantly it is the site of RNA synthesis. It is the site of RNA synthesis. That is, DNA makes DNA to RNA, and RNA synthesis occurs down here during chromosome puffing. Hence the right choice is 5. The answer is C. The answer is C for chromosome puffing. Now normally this is a sort of question which normally students get confused on, because we predominantly we think it is protein synthesis. It is not protein synthesis, but only one single component which is termed as RNA synthesis. So chromosome puffing, Balbiani rings and RNA synthesis, these are the sort of an associated terms which any particular student need to exactly understand whenever the term chromosome puffing comes in with relation to any sort of an objective bit. Question number 6 states, who proposed the chromosome theory of inheritance? The chromosome theory of inheritance was proposed by, the options are Sutton, A, B is Boveri, C is Sutton and Boveri, and D is Correns. Now, when you take the chromosome theory of inheritance, it was the first evidence which has been proposed by Sutton and Boveri. They were classical geneticists who had proposed about a century back the chromosome theory of inheritance, indicating that chromosomes are the vehicles of inheritance. Or chromosomes predominantly are the determinants and the carriers of genetic information from one generation to another, and again the chromosomes bear the DNA. They bear the sort of a DNA. Whereas when you take the last choice, Correns is associated with extra chromosomal inheritance. So the right answer is C. That is, both Sutton and Boveri are associated with the chromosome theory of inheritance. This comes under transmission genetics, as well as it's an example of a theory under classical genetics. We go for question number 7. This is a baseline question. Now question number 7 asks you, where is the gene for color blindness? The gene for color blindness in man is located on, because it is sex linked, it is predominantly located only on the X chromosome. A choices X chromosome, B choices Y chromosome, that is the maternal chromosome. C is it either X or Y, or is it D is both X and Y chromosome. As you understand, it is sex linked, and thereby the color blindness is basically present on the X chromosome. It is present on the X chromosome, and as implied, man carries the gene, whereas the woman with the Y chromosome are basically carriers. They are only the carriers, but the gene for color blindness, is associated with man or with relation to the X chromosome. Hence, the answer is A for color blindness. Now, question number 8 says, a colorblind daughter may be born if the choices are A, the father is normal, mother is colorblind; B, father is colorblind and mother is normal; C, father is normal and mother is a carrier; and D, the father is colorblind and mother is a carrier. As implied through several studies on the genetics of colorblindness, we always understand that the gene for colorblindness is transmitted by the father. The basic principles in the genetics of colorblindness is father has the gene for colorblindness. And the mother is the carrier. Mother is the carrier. And thereby, when you take the daughter is also a colorblind carrier, whereas when you take the son inherits the gene for colorblindness. Now, among the 4 choices, choice 8, the correct answer is D, which says the father is colorblind, and mother is the carrier. And in view of these 2 statements, the correct answer is D. The correct answer is D. So this forms the basis for this is the norms or the basic rules for colorblindness. Gene from the father, carrier is a mother, daughter is also a colorblind carrier, and when you take the son, the gene, he has got the gene for colorblindness. So naturally, in the pattern of inheritance, 50% of the girls or the daughters are the carriers, and 50%, that is the sons, inherit the gene for color blindness. We go to question number 9. Question number 9 asks, a normal woman is married to a colorblind man. We have the man who is colorblind. The children are expected to be A, all normal. It's not the case. B, 50% of the sons are colorblind. Yes, to a certain extent it is right. But C, all the daughters are normal, but they are the carrier, whereas all sons are normal phenotypically as well as they are genotypically. Genotypically they carry the colorblind gene. And D is 50% of the daughters are colorblind. So when you analyze the choices, the nearest probable choice here is all the daughters are normal. Daughters are phenotypically normal. But they are the carriers. They are the carrier of the colorblind gene. They are the carriers, whereas all the sons are normal phenotypically as well. Sons are also phenotypically normal as well. So both the daughters and sons are phenotypically normal, and their also sons are also genotypically normal. So when you say they are genotypically normal, it means they are carrying the gene for color blindness. They have the gene for color blindness. Hence, among the 4 choices, the best choice is answer is C. Answer C, which indicates phenotypically both of them are normal. Genotypically the sons too are normal, but they carry the gene for color blindness, and in the option the daughters are also the carriers of color blindness. So C of the C bit correctly supports the concept of color blindness and its pattern of inheritance through the male as well as to the daughter as well as to the sort of a sons. Next we go to a very simple question, question number 10. Which of the following disease is sex linked? Is it color blindness, or is it malignancy, or is it hepatitis, or is it leukemia? Now among the choices, malignancy is cancerous, cancer is not sex linked. Hepatitis is a metabolic disorder, leukemia small cell cancer, and the right sort of an answer is A, which says colorblind is sex linked. So we understand that color blindness is sex linked to the male. Thereby the correct answer for 10 is answer is A. Question number 11 requires a little bit of introspection. A color blind son is born to normal parents. When you have normal parents and to them you have a color blind son, it shows the choices are A, the father was heterozygous for color blindness, B, the mother was genotypically homozygous, C, the mother was heterozygous for color blindness, D, both parents carry a recessive gene for the disorder. Now we will follow the pattern of elimination. Both the parents do not carry a recessive gene for the disorder because mother does not carry the gene. Then C, the mother was heterozygous for color blindness. It could be possible that the mother is a carrier and the colorblind gene is inherited from her father. So the son gets the sort of a gene from the grandfather. Whereas when you take choice B, the mother was genotypically homozygous. The mother's gene does not affect color blindness at all, and the father was heterozygous for color blindness. Father doesn't come into the sort of picture because primarily it's just a sort of a normal parent, wherein exactly it is said that the parent does not have the sort of a colorblind gene. So the colorblind gene which the son has inherited via the mother comes from the grandfather. Hence for the 11th bit the choice is the mother was heterozygous. You take the mother is heterozygous for the gene of color blindness. So it principally says from the grandfather it comes to the mother, and the mother is heterozygous over here, and from the mother it goes to the son who is born to parents who are normal. So the son gets the colorblind gene from the grandfather. Hence the answer to this is C for color blindness. Now question number 12, again a simple but a direct question. Where are sex-linked characters? Sex-linked characters are those which A, they are related to the sexual physiology, B, the genes of which are present on the sex chromosomes, that is the allosomes, C, which appear either in male or in female, that is it is sex limited, D, which is controlled by sex hormones or pheromones. Now whenever we are dealing with relation to sex linkage, it means a character is associated with the sex chromosomes. the sex chromosomes of X or Y. Sex or Y. It is not related to A, sexual physiology, because we are dealing with relation, when we say sex linkage, we are dealing with relation to inheritance. As you understand, the science of genetics is the science of inheritance and the transmission of characters from the parents to the offsprings, as well as the variations. Choice B says genes of which are present on the sex chromosomes. So when you take the sex chromosomes, we have certain characters, and these characters have genes which are expressed, and the genes are related to the sex chromosomes. So the part B, that is the choice B, the answer B is right, while we eliminate which appears either in male or in female. If a character appears either in male or in female, it is termed as sex limited. So sex limited is either X or Y, which is not so here, and which are controlled by sex hormones. They are never, the characters are not controlled by sex hormones. So the right answer seems here. We have the character which is present on the sex chromosomes on the genes, which can be there, which can be X and Y. So the answer is B. Question number 13 says the chromosomes other than sex chromosomes. Chromosomes other than sex chromosomes are called as A is autosomes, B is heterosomes, C is karyosomes, D is none of the above. Normally chromosomes other than sex chromosomes are termed as autosomes. Now the meaning of the term autosomes, autosomes are somatic chromosomes. Somatic chromosomes which are responsible for somatic characters. So the correct answer for 13 is answer is A. Answer is A. Going to question number 14. A colorblind man marries a normal woman whose father was colorblind. That is, the grandfather was colorblind. What percentage of the children is expected to be colorblind? The choices are 25%, 50%, 75%, and D is 100%. As implied even in the last problem, we always understand that grandfathers have the gene for inheritance. Normal parents also, normal parent male has got the gene for inheritance. Mothers are the carriers of inheritance. And when a normal man, when a normal woman marries a colorblind man, the probability of the male and the female offsprings are 50:50%. And normally for all the daughters, it is expected the daughters are the carriers for colorblindness. Hence, the probability or the percentage of the children to be expected to be colorblind, that is, the male, is 50%. So we have 50% are colorblind, that is the sons, and 50% are colorblind carriers, that is, we have the daughters. Hence, for 14, the answer is B. Question number 15 says, in 4 o'clock plant, that is Mirabilis jalapa, normal leaves with an A gene and variegated leaves with B occur in different plants. It states that you have got normal leaves as well as variegated leaves due to the expression of two different genes, A and B. If B male, that is a variegated male, is crossed with A female, that is with normal leaves female, the hybrid has normal leaves. But when B female, that is variegated female, is crossed with A, that is normal male, the hybrid has variegated leaves. It is a case of A, mutation, B, cytoplasmic inheritance, C, complementary genes, D, supplementary genes. So many a time, if you take Mirabilis jalapa, Mirabilis jalapa is called the 4 o'clock plant. Because it flowers at 4 p.m. And if you take Mirabilis jalapa, we are looking at leaves. The leaves could be normal or it could be variegated. When you say variegated, it means they are colored. They are colored in various shades of green, yellow and white. Variegated leaves are also termed as mottled leaves. Now, mottled leaves are inherited, as you understand from the problem. In the problem, it says there are different sort of combinations which occur. So, in an objective bit, we don't have to go through the pattern of inheritance, but we will have to exactly know that there could be two variations, and these variations are inherited. So, because these variations are inherited, choice A, that is mutations, is wrong, because mutation is an unprogrammed change wherein there is a change in the chromosome number or the structure, and mutations need not be inherited. Choice number C indicates it is due to complementary genes. When you say complementary genes, two genes come together and they determine a particular sort of a character. So, complementary genes, and choice D, which says supplementary genes, when two genes add up to determine a particular character, these two patterns of inheritance is because of inter as well as intragenic interactions. That is, if you take a choice of capital A dash B dash, A and B can interact together to determine a particular character. Similarly, you take A and small bb, it can determine a particular character. Same is the choice with small a and capital B, they also can determine a particular character, or you have a small a and a small bb which can determine a particular character. So, these are termed as examples of both complementary as well as supplementary genes. But when there is an inheritance pattern, first thing is, when there is an inheritance pattern where there is a normal leaf and a variegated leaf, and there are variations due to crossings. The variations due to crossings is due to the presence of cytoplasmic inheritance. It is due to cytoplasmic inheritance. So, cytoplasmic inheritance or maternal inheritance is a pattern of inheritance wherein the genes for coloring, that is the chloroplast genes, get mutated, and the chloroplast genes are transmitted through the female line. So, any transmission via the female line related to pigmentation as evident in Mirabilis jalapa, or as evident in terms of the character of variegation, is an example of cytoplasmic inheritance. So, the correct answer for bit number 15, question B, the correct answer is B, which says that cytoplasmic inheritance is the pattern which is normally associated with the 4 o'clock plant. Question number 16. It states, who discovered sex-linked inheritance? Sex-linked inheritance had the meaning of the term sex-linked inheritance, inheritance pattern which is present due to the presence of an X gene or a Y chromosome, and this has been studied in Drosophila by a very important scientist whose name is T.H. Morgan. Morgan studied fruit flies, and he established the pattern of sex-linked inheritance, whereas Bridges is associated with crossing over, McClung is associated with sex chromosomes, and Wilson and Stevens are associated with biological diversity. So, the most nearest or the best answer is T.H. Morgan. T.H. Morgan is always associated with sex-linked inheritance. So, Morgan, Drosophila and sex-linked inheritance are major discoveries in the field of classical genetics or transmission genetics. Hence, the correct answer is A. Correct answer is A for sex-linked inheritance through Morgan. Question number 17 asks, color blindness in which of the all colors appear grey is termed as monochromatism or dichromatism or protanopia or deuteranopia. Now, whenever we see color blindness is a sort of a character wherein you can't distinguish between red, blue and green. Predominantly, anybody who is color blind only looks through shades of grey. They can only perceive shades of grey, and there is a dimming between the blue and the green colors. So, when there is a dimming between the blue and the green colors and only one color is expressed, it is termed as monochromatism. So, monochromatism or ability to visualize only the grey color instead of the blue-green color is termed as color blindness. Now, this is a very important test for the selection of pilots as well as railway guards. And people who work on computers. So, a screening test for color blindness for selection is apt for the selection of pilots, railway guards and computers. And monochromatism is the basis. Hence, the correct answer is A. Question number 18 asks, in Down syndrome, the chromosome number in each cell of the body is. So, the choices are 45, 47, 48 and 49. As implied, always we understand that in the case of the Down syndrome, always the chromosome complements are 47. Hence, the answer is B. Question number 19 asks, Down syndrome is caused by the presence of an extra chromosome number. The extra chromosome number is on X chromosome, Y chromosome, on chromosome 21 or chromosome 22. The right answer is chromosome 21. On chromosome 21, we have the presence of an extra chromosome. Hence, the right answer is C. We are going to question number 20. The child afflicted with Down syndrome has A. Flattened nasal bridge, open mouth with protruding tongue, or choice B is small forehead, bulging eyes and raised nasal bridge. Choice C is habitual open mouth with long protruding tongue, bulging eyes and a small forehead, or D is large forehead, raised nasal bridge and long included tongue. Now, when you take Down syndrome, any child afflicted with Down syndrome has A. Flattened nasal bridge, the nasal bridge between the mouth and the forehead, this nasal bridge is flattened. And same is the case between the mouth. Supposing we have the mouth over here, even over here it is flattened. So they have a flattened nasal bridge and always an open mouth. The mouth is open. And we have a protruding tongue. So, in the case of a Down syndrome, flattened nasal bridge, protruding tongue and mouth open are the three specific sort of a characters by means of which you can identify the Down syndrome. Hence, the correct answer is A. Next, we go to question number 21. Question number 21 says, a person having Klinefelter syndrome. A person having Klinefelter syndrome is characterized by choice A is they are male with some secondary sexual characters of a female. B, they are female with some secondary sexual characters of the male. C, having both male and female sex organs. D, female internal sex organs and male external sex organs. So, when you take Klinefelter syndrome, Klinefelter syndrome which has been discovered by Klinefelter, as you could recollect, the last choice we go by the process of elimination. The female internal sex organs and male external sex organs does not have any logic, so the statement number D is wrong. Having both male and female sex organs, it's not they are not a sort of a neuter person. And again, when you take Klinefelter syndrome, it is not associated with the female. A female with some secondary sexual characters of the male is obsolete, it's not correct. Hence, the right choice is, whenever we are dealing with Klinefelter syndrome, we understand it is a sort of syndrome which is caused on the male with a few of the secondary sexual characters of the female, which is seen in the form of a pitched shriek voice and development of breasts, and again maybe long hands and a bit of webbing on the tongue, webbing of the neck and talkativeness. So, among these, the near about secondary sexual organs are these. Hence, the choice for 21, the answer is A. Answer is A. Question number 22 says, in human beings, 45 chromosomes with a single X or a XO abnormality is the causes due to A. Down syndrome, Klinefelter syndrome, C is Turner syndrome and D is Edwards syndrome. So, when we have 45 chromosomes, that is, we primarily have 45 chromosomes and the 45 chromosomes with an X, single X, or it is given as XO. When you say O, it means Y is absent. Y is absent. And this is an abnormality, because there is a presence of an extra chromosome. Now, this is the case in the case of the Turner syndrome. Turner syndrome normally have an X or an XO. Hence, the correct answer for this is C. Answer is C. Question number 23 is a direct question. Down syndrome or Mongolism is due to the presence of an extra sex chromosome, that is an extra X or Y. B, extra 21st chromosome. 21st chromosome has got an extra one more add-on chromosome, or is it C is an extra Y chromosome, that is you got a double Y, or the choice D says there is a deficit sex chromosome, that is it minus X or minus Y. We always understand when you take Down syndrome, or it is Mongolism, idiocy, it is always due to the presence of an extra 21 chromosome, extra 21st chromosome. Hence, the answer is B. Question number 24 is a direct question. Genes for color blindness in humans is carried by the mother, the father, both, or is it an abnormal sex? Now the question is little tricky. In a sense, genes for color blindness genes is predominantly present. When you say it's carried, there is a high probability that you will write it is carried by the mother, but it specifically says genes for color blindness. So the gene for color blindness in humans is carried, primarily it is transmitted from the father. The more appropriate sort of a word would be is it is transmitted from the father, and the mother is a passive carrier, and the mother is a passive carrier. So hence, for this particular bit, the answer is B. The answer is B, which says the father is the carrier of the sort of a gene. The carrier of the gene is the father. Question number 25. Again a direct question. Color blindness is due to recessive allele in females, dominant allele in females, dominant allele in males, or is it a recessive allele in males? As implied, color blindness is also always due to homozygous condition, and it is because of recessive genes, and it is present in the males. Hence, because of the presence of a recessive allele, the correct answer is D. So color blindness is a recessive gene, and the correct answer is D. It is a recessive allele in the males.